Well, dear students, let us discuss the next topic. That's the life cycle of Taenia solium. I have already told you in the previous lecture that the Taenia solium is digenetic, means it completes its life cycle in two hosts. The primary host is male, secondary host is pig and the other animals. Uh, the life cycle of Taenia solium is indirect. That means it involves various larval forms, but none of the larval form is free living. So all the larvae they will grow in the secondary host. They will not grow outside the host. They don't have any free larval stage. Self-fertilization takes place because single tapeworm is present in one host. One host means primary host, that is main. Primary host is the host where sexual, yeah, or you can say the adult organisms are present, where the uh, sexual mode of reproduction takes place. So adults of the Taenia uh, occurs are present in the human being. So the human being is the primary host. This self-fertilization is sometimes also called as cross-fertilization. Why? Because of this reason. That the copulation takes place due to folding of strobila. Because if you have remembered, that the Taenia solium has about 800 to 900 strobila, and each strobila is a separate unit having separate reproductive system. So what happens that when one strobila fold upon another strobila, then the common gonopore of the two mature proglottids come in contact, and as a result, the penis of one is inserted into vagina of the other. So, if you are considering each strobila or each segment as a separate subunit in terms of reproductive system, so then it is cross fertilization because the uh, sperms of one segment are going to fertilize the egg of the other segment. So in that case, it is the cross fertilization. But if you look at in terms of individual, because only one individual is involved, so it is called the self fertilization. I have already told you that sperms remain stored in the seminal receptacle. Why? We have seen that in the previous lecture that the male reproductive organs develop earlier and female reproductive organs develop later. Though within the same segment, both the reproductive systems are present. And when male develops earlier, this is called protoentry. So, because of the protoentry, when the sperms of the male reproductive system is mature enough and the female has not matured now, then what will happen that there is a need that the sperms should be stored somewhere so that when egg will mature the fertilization may take place. So the sperms are stored in the seminal receptacle. Seminal receptacle is the part of the female reproductive system, not the male reproductive system. Till the ova is released, the two finally, and you might have remembered that in the previous lecture we have shown you the fertilization duct where both egg and sperm meet together. This is because it is taking place within the reproductive system. It is called as internal fertilization. Then the fertilized egg passes into ootype. In the ootype, it gets yolk, from the vitelline gland, which meets the ootype through vitelline duct and enclosed in a shell or chorionic membrane. So you can see over here. This A is the fertilized egg cell or zygote. So this fertilization takes place in the fertilization tube. Then this B is a capsule formation in ootype. What happens when they reaches the ootype? They start getting the yolk from the vitelline gland. So this is the yolk cell formed around the developing zygote. In addition to it, the egg shell or the chorionic membrane is also formed. Okay, so this is after fertilization that these two things are added to it. One is yolk cell from the vitelline gland, and then the egg shell or the chorionic membrane. Then from C to E, they are the oncosphere formation in the uterus. So you know that after taking these two material in the ootype, the this very structure, this zygote now moves to the uterus of the gravid proglottids. In the gravid proglottids, these changes take place. What happens over there? That the egg cell or the zygote divides unequally into two types of cell. One is large in size, other is small in size. The larger one is called as megameres, and the small one is called as micromere. So its name is micromere. Both these are called the embryonic cells. Rather, no, sorry. The micromere is called the embryonic cell. Then what happens? That this micromere further divides into many small cells like this. These are the micromeres. And the large mesomeres. So two types of cells are formed from the division of the this embryonic cell. One, the smaller cells are called the micromeres, and the medium type of size cells are called the mesomeres. Then the megamere also divides into many larger cells called the megameres. So you have three sizes of the cells. The megameres, the largest one. The smaller to the megameres are the mesomeres, and smaller to them are the micromeres. Mesomeres and micromeres are formed by the division of embryonic cell, whereas megameres are formed by the division of megameres. Then what happens that these large mesomeres, no, first see this, the micromeres, the small micromeres further divide and form the morula. Morula is the mass of the cell in the early development of the embryo. So morula micromeres are there. Then around them a layer is formed which is called as inner embryonic membrane. This is called as inner embryonic membrane. It is formed of it is also called embryophore secreting mesomeres. These are the embryophore secreting mesomeres because they will secrete embryophore later on. This layer is formed from the mesomeres. So these medium type of cells will develop a layer around the morula, which is called as embryophore secreting mesomeres or inner embryonic membrane. It is a hard layer. Outer to them, the outer to them, the megameres will arrange into a layer. They will take up the yolk from the this yolk cells which were present over there in the periphery, and they will form the yolky megameres. So the yolk from the yolk cells is taken up by the megameres, and the megameres have arranged themselves into an outer membrane. This is called as yolky megameres cell membrane or the nutritive envelope are also called as outer embryonic membrane. So this is the outer embryonic membrane. Even after taking the yolk from the yolk cells, few yolk cells may remain outside them. Then what happens that within within that structure, this morula then develops into this morula then develops into this is the morula, and around the morula, this is the embryophore. Who has formed the embryophore? This layer, which is what's called as embryophore secreting mesomer or inner embryonic membrane. So inner embryonic membrane has secreted the embryophore, and a eggshell is present outside it, and few vital end cells are still present having the yolk inside them. This structure is called as young oncosphere. Oncosphere is basically the first larval form which will be formed after the fertilization. So this is the development of oncosphere larva. This is the young oncosphere. This young oncosphere will develop into the mature oncosphere. What will happen? That this morula, whole of the morula will turn into the oncosphere or the hexacanth larva. Oncosphere is also called as hexacanth larva because it has the six hooks or three pairs of hooks. This is the embryo, hexacanth embryo, you can say. And outside to it, again the embryophore is present. You can see this is the first membrane or the embryophore, and then outside it, as usual, eggshell is present. Eggshell is also called as chorionic membrane or second membrane. So first membrane around the embryo is the is this embryophore, and the second membrane around the embryo is the chorionic membrane or the eggshell. This hexacanth, besides having the hooks, also have the penetration glands. What is penetration gland? When this larva will penetrate the secondary host, then in order to penetrate inside the body of the secondary host, it requires certain enzymes for digesting the wall or the muscles of secondary host to get into the muscles. So the enzymes are packed in the penetration glands. Oncosphere loses its shell membrane, so the embryophores become outermost. What will happen later on? This eggshell will be dissolved, and the embryophore, the first membrane, will become outermost because it will lose later on the eggshell. Still up to this development is taking place within the gravid proglottids. And gravid proglottids means still in the primary host, in the intestine of primary host, the tinea solium in the gravid proglottids may have 30,000 to 40,000 oncospheres packed in embryo in the packed in its uh, gravid proglottids. Okay, then gravid proglottids now detach in group of 4 to 5. What will happen when development up to this stage has taken place? The gravid proglottids will detach from the posterior most end, which was called apolysis. Then 4 to 5 gravid proglottids pass out with host feces. They will come outside the body, rupture on the ground. Gravid proglottid will get ruptured on the ground, and oncosphere larva with this structure will get released from the gravid proglottids. Then what happens? Infection to secondary host cell takes place when the oncosphere larva lying on the ground will be ingested by the secondary host. Say, for example, the pig. When pig will eat or take up the feces of the primary host. Sometimes what happens that autoinfection may take place. That means the the gravid proglottids may may be may get or you can say the oncosphere larva may infect the primary host again. How it will happen? It is called autoinfection. It will happen by this in primary host due to reverse peristalsis. Because normally what happens when the gravid proglottid will detach from the tinea solium, they must pass out with the feces of the primary host. The passing out of the feces is because of the peristalsis, the movement of the intestine, which excrete out the excretory material or the excreta. So, if the peristalsis or the movement of the intestine will get reversed due to some reason, then what will happen? The detached proglottids shall be taken backward from the large intestine into the stomach, where oncospheres shall be liberated because of the digestive enzyme of the stomach, and the oncosphere may infect the primary host. It will enter into the wall of the primary host. This is called as autoinfection. Now, say for example, the oncosphere has come out along with the feces of the primary host, then it will migrate in the secondary host. In the digestive system, oncosphere shall lose its surrounding membrane, first membrane as well as the second membrane. I have already talked about by the action of acidic and alkaline juices of the intestine of the secondary host. So, in the intestine, the enzymes will act upon the both of the membrane. As a result, this structure, the oncosphere, shall become now liberated from the earlier membrane. Earlier, it was having these two membrane, the first membrane embryophore and the second membrane eggshell. Both of them shall get dissolved by the enzymes, and hexacanth embryo may become free within the intestine of the secondary host. Then what will happen? Free hexacanth, with the help of hooks and penetration gland, bores the intestinal epithelium. So in the secondary host, they are present in the lumen of the intestine. With the help of hook and penetration gland, the hexacanth larva will bore the epithelium to reach the submucosal blood. So if you remember the histology of the intestine, so intestine have epithelial layer uh, in the surrounding the lumen, and then just beneath the epithelium you have submucosal blood or lymph vessels. So the larva will reach to the blood or lymph vessels. They, after reaching the blood vessel, it will shed off the hooks, because the purpose of hooks was to attach with the wall of the intestine. No, it has, after getting attached, it has entered the blood vessels or the lymph vessel. There is no function of hooks, so hooks shall be shed off, and with the help of blood, this larva shall be carried via hepatic portal vein to the liver. The liver, hepatic portal vein is the vein that is coming from the wall of the intestine to the liver. So along with the blood, it will reaches the liver. Then from liver, it will reach along with the blood to the heart, and then to the arterial circulation, where it will circulate whole of the body. Finally, it reaches the muscle. So from the arterial circulation, along with the blood, it will reach the muscles of tongue, shoulder, neck, thigh, heart, where it settles to develop into a bladder worm or cystic. So once it will reach there in the muscles, then the next larva will be formed, which is called as cystic. This is the cystic. How the cystic will be formed? You can look at this. That the because the hexacanth larva at this stage has lost the hooks, there is no hook. Invagination shall takes place, and because it was traveling in the blood, so whole of the bladder of the this uh, the larva shall remain filled with the blood of the primary host. This is the invagination. This is the wall of the bladder. This is outer cuticle and inner mesenchymal or germinal layer. Two walls basically. The outer cuticle and the inner is called mesenchymal or germinal layer. Then what will happen? That in pig's body, cysticercus lead an inactive life for several years. So in the muscles, it will develop into cysticercus, will remain as such for several years, even dies after some times, but it develops into adult only when it will be ingested by human host. So when human host up we shall take up the meat of the pig, then this cysticercus may get into the human being. No, we were discussing the development of cysticercus. Then later on, what will happen? That scolex will be formed within this invagination. So scolex, you might have remembered, it is having suckers, the prososcolex along with the hooks, and again rest structure is same. That is the two layer outside called the outer one is cuticle, inner one is germinal layer, and the bladder is filled with the blood of the primary host. Now, infection to primary host. What will happen? Up to this stage, all development has taken place within the muscles of the secondary host. What will happen? How it will reach to the primary host? Now, infection by eating uncooked measly pork. Is measly pork. Pork means the flesh of the pig. So, if the flesh of the pig shall be taken by the uh, primary host, that too in uncooked manner, then the cystic will get the chance to enter into the intestine of the primary host. And inside the intestine, Prosocolex evaginate. Earlier it was having downward position. Now it will evaginate. Because of evagination, this Prosocolex will come out from the invagination, and with the help of the hook, it will attach to the intestine wall of the primary host, and neck shall begin to proliferate and bladder is digested. So from the neck, the proglottids will be formed, and slowly and steadily, whole of the bladder will get exhausted. This is the summary you can say uh, in the intestine of the human being. This was the adult tapeworm, the gravid proglottid, having the uh, uterus filled with the eggs or the developing embryo shall come out along with the feces. Then outside, when it will come outside, gravid proglottid will get ruptured. Inside it, the developing embryo will be released. These embryo will be once taken up by the pig, then inside the intestine of the pig, they will shed off their outer layers, and the hexacanth larva shall be liberated, which will attach with the muscles of the intestine. As a result, it will enter into uh, it will penetrate the epidermal layer of the intestine, will shed off the hook, will become the cysticercus, and when the muscle part along with the cysticercal larva shall be taken up by the human being, it will again go into the intestine of human being, where it will attach with the wall of the intestine of the primary host or the human being, and the development of the adult shall takes place by the proliferation of the neck region, and then the formation of the adult as a whole. So far as the pathogenesis is concerned, pathogenesis means manner of development of disease. So you have different types of diseases due to the infection of the Taenia solium. One is called cystidiasis. What is cystidiasis? Because you must remember that this was the cysticircus, which was the infective stage for the human being. So, if the problem in the primary host, the disease in the primary host, shall takes place by ingesting the cysticirci, so it can be called as cystidiasis. So I have already told you cysticirci shall get into the primary host by taking the uncooked measly pork. Then teniasis may takes place. The symptom of teniasis is that once it will be taken inside the intestine, it will develop into tinea solium, and once the adult tinea solium will be formed in the intestine of the primary host, then there will be abdominal pain, there will be nausea, means feeling of sickness, the blood loss, anemia, there may be an increased appetite, there may be indigestion, increase of eosinophil cells in the blood, then nervous disorder like epilepsy may occur. It will be caused by the toxin produced by the tinea solium, and because of the mechanical irritation in the intestine, which will initiate the reverse peristalsis, it may lead to the autoinfection. So the reverse peristalsis normally does not take place in the primary host. It will take place because of the of the irritation caused by the tinea solium in the primary host, due to which autoinfection may take place. Usually, a single tapeworm is found in the found to parasitize a host, because it will not allow the second parasite to get into develop into the intestine. So it provides the immunity against the fresh infection. So only single tapeworm may be found in a host. You may not find two Taenia solium at a time. Then the third type of disease may be called a cysticercosis. Say, for example, due to autoinfection, the cysticerci from the stomach have entered into the muscles of the primary host, then the cysticercosis may develop. This is more dangerous, as encystment of bladder may takes place in the host voluntary muscles, and once the cysticercosis will develop into the or will take the position in the voluntary muscles like the cardiac muscles, the liver, the eye, the brain, and the patient, uh, other other cells, the patient shall show epileptic behavior, and all these functions of all these muscles may get disrupted, and it will only after removal of cysticerci from these delicate organ shall be the remedy. But because these are so delicate organ that it is not always uh, good to operate them, so it is extremely difficult to get cysticerci or the cyst developed because of the tenosolium in these organs. So this is more dangerous, because you cannot remove from these delicate organ this very cyst. Then there is a treatment that you can give the anti-helminth drugs, but these drugs like camoquine, carbon tetrachloride, oleoresin, quinacrine, antiphane, dichlorophane, vomacin, all these are present. They will remove the strobila from the intestine, but they will not do anything to the cyst if it is developed in the muscles. However, scolex can be removed by surgery. So you can see scolex may remain attached, strobila will be removed. So it means again the tinea solium will be formed, because the neck region next to the scolex shall give rise to the more strobila, and then may regenerate the parasite in the primary host. So prevention or the prophylaxis is that you must not take the uncooked meat. Why we are taking uncooked meat? Sometimes we check the meat whether it is cooked or not by tearing the meat, uncooked meat, with the help of our teeth. In that very chance, it may get into the primary host, mm, then the infection may take place. So feces of infected person, if you are finding that a person is infected, they should not they should be properly disposed of. That means he must not be allowed to put his feces outside, so that a secondary host may get infection, and again the cycle will be completed. So they must be properly disposed of and destroyed. Thanks. This is all about the life cycle of the Taenia solium, the pathogenesis, and the prophylaxis.